Good everyone. So we're going to talk now about some parts of the brain and evidence over how they are different in antisocials than in controls. I'm going to split it up in terms of the structural evidence and then the functional evidence. Okay, keep that distinction in mind. If there's a question in the exam, it might be specific, and whether it's asking you about structural or functional, you have to make sure you give the right piece of evidence. Also, you've hopefully looked at the essay by now. There's 2 essay questions to choose from, and one of them refers to structural functional differences between reactive and proactive criminals. The parts of the brain that I'll be discussing are pretty much the same, mainly the prefrontal cortex and the amygdala. But we're going to look at structural differences today and then functional differences on Tuesday. So first of all, there's a number of methodologies we can use in order to gain structural or functional information on the brain. We can use EEGs, which in comparison to the others, give us a pretty simple form of data, but it tells us the electrical activity that's happening in the brain. We can do some comparisons, for example, between when you're awake and when you're asleep. An event-related potential is the same as an EEG, but it's in response to a particular event, a particular stimuli. A PET, a positron emission tomography, is when we put a tracer into the bloodstream, and then it allows us to monitor blood flow within the brain, so we can see what parts of the brain are currently active. Okay, looking at what parts of the brain have blood flow currently, which tells us something about functionality. An AMRI gives us high-resolution images of the brain, and it is high resolution. It allows us to see with precision the volume of particular brain areas. So maybe we could do comparisons. You know, is this part of the brain structurally different than normal? Is it larger or smaller than what is typical? And an fMRI is really the best of both worlds. It gives us both functional and structural information. It gives us those high resolution images, but also tells us about the current activity within the brain. So what parts of the brain are currently active. Now, we've talked already about some prefrontal cortex findings, in that prefrontal cortex dysfunction is a strong correlate with antisocial behavior. I'll tell you about an interesting study in which Adrian Raine wanted to look at the difference in prefrontal grey matter volume between controls and antisocials. He wanted to recruit antisocials from the community rather than within prisons. So where do you best look to find antisocial individuals in the community? Well, he did a lot of scouting, and it turns out the best place he found was temp agencies in Los Angeles. Okay. And so recruiting from temp agencies in Los Angeles, and there's many reasons as to why one might be working for a temp agency, but certainly one reason could be a difficulty holding down a job. Okay. He recruited participants and then looked for signs of an antisocial personality disorder to make diagnoses. So we had a group of controls, all recruited from the same place, all recruited from temp agencies in LA, but a group of controls recruited from there who don't meet the criteria for an antisocial personality disorder. We have a group who have an alcohol issue, a substance dependence issue, that don't meet the criteria for an antisocial personality disorder, and then a group that do. Okay. As a separate measure, we also use the psychopathy checklist, okay, to see if the difference in psychopathy from group to group is what we would expect, okay. The psychopathy checklist measures psychopathic characteristics out of 40, right? For research purposes, 25 is the cutoff point, typically, to be classed as a psychopath. In other justice systems like the UK, 25 is also the cutoff point. Here in the US, it's 30, because the consequences of being labeled a psychopath are more severe. There's the death penalty in this country, there's harsher prison sentences, so on. And if you are classed as a psychopath, you're more likely to get the death penalty and more likely to get a harsher prison sentence. So 30 is the cutoff point for the criminal justice system here. So the controls have an average score of 14. It's not a normal score from a community sample. From a community sample, we would expect typically 5 out of 40. Okay, so it's above average for the community. But keep in mind the sample that we're gathering from. The substance abuse group score of 20, so it's higher. It's not at the cutoff point for psychopathy, okay, but it's higher. And then there's group of antisocial personality disorder, significantly higher, right? It's between 28, 29. It's above the cutoff point for psychopathy for research purposes, not quite at the cutoff point for the legal purposes, but it's close to it. But it's in the direction that we would expect. And we'll talk about psychopathy later in the course, but you can think of it for now as a more extreme personality disorder, a more extreme form of antisocial personality disorder. Okay. So this group of antisocial personality disordered individuals, what we want to see is how their prefrontal grey matter volume differs from the other groups. Who is this group? Who is this group of 21 individuals? What kind of characters are we dealing with? Well, 43% of them say they've raped a woman. 53% of them say they've attacked a stranger, causing bruising or bleeding. 29% say they've committed an armed robbery. 38% of them say they've fired a handgun at a person. And 29% of them say they've killed someone or attempted to kill someone. So that's the kind of antisocial behaviors we're talking about in relation to this antisocial personality disordered group. Okay. Now, overall, this group of antisocials had 11% reduction in prefrontal grey matter volume. Okay, you can see it's significantly different than the other two groups, than the controls or the substance-dependent group. Okay. They also had reduced atomic activity in the atomic nervous system. And I'm going to talk about that in a future lecture. I'll come back to this study. We'll talk about the atomic nervous system in that lecture. For now, I just want to talk about the structural differences in the prefrontal cortex. So they have less prefrontal grey matter volume. Okay, that's on average in terms of the prefrontal cortex. But we can be more specific than that. We can narrow down in terms of what parts of the prefrontal cortex are most different in this group of antisocials in comparison to the controls. If you imagine taking a slicer through the prefrontal cortex, you would end up with an image that looks like this. Okay, and then we can break down the outer area, the frontal gyrus, into subsections. Okay, so we can compare those subsections within the antisocials to the controls to see what subsections are most different. Okay, this is one way of splitting it up. So you could have the section at the top for the right and the left hemisphere, which we are calling the superior frontal gyrus, superior for on top. Okay. The middle frontal gyrus on the next part down, if you like. The inferior frontal gyrus, inferior for lower. Okay. The orbital frontal gyrus. Okay. And then on the bottom, the rectal frontal gyrus. Okay, and then we can look at these subsections to see which parts are most different between the antisocials and controls. So what we have here is the average difference for the antisocials in their prefrontal grey matter volume in comparison to the controls based upon each part, okay, of the frontal gyrus. So you can see it's color coordinated, right? That's the superior frontal gyrus, that's the middle frontal gyrus, inferior, all the way around. Okay. If it's blue, it's a non-significant reduction. Okay. If it's red, it's a statistically significant reduction in comparison to the controls. Okay. So what part is most different? Well, both sections of the middle frontal gyrus, right? between 11 and 20% reduction in thickness in comparison to the controls. It's pretty substantial on both hemispheres. Also, the orbital frontal gyrus, again, on both hemispheres, is 9 to 10% lower in thickness than the controls. Again, a substantial difference that's statistically significant. For the rectal frontal gyrus, it's only the right hemisphere, okay? 16.4% reduction. The others are non-significant, right? There's no statistically significant difference between the controls and the antisocials for the superior frontal gyrus, or for the inferior frontal gyrus, or for this side of the rectal frontal gyrus. Okay. So that's the parts of the brain in particular, okay, that are reduced in thickness for these antisocials. Now, if we look more closely at those particular parts that were most different and see what we know is associated with those parts of the brain, we can try to see why that might be the case, why it is that these parts of the brain are reduced in thickness. Does it mesh with the broader research? So one was the middle frontal gyrus, right? That was one on both sides that was significantly reduced in the antisocials in comparison to controls. But what has the broader research told us that this part of the brain is associated with? Well, it's associated with awareness during fear conditioning. That would make sense. We already know that antisocials, criminals, don't fear condition as well as typical people do. Empathy to pain stimuli, well, they're not the most empathetic individuals, right? You've just looked at their criminal histories, right? Nearly half of them committed rape, nearly a third of them had committed murder or attempted murder. Moral decision-making, again, not surprising that that would be impaired, right? Response inhibition, well, we know that antisocials are more impulsive, okay? Choosing to delay immediate gratification, okay, in order to get, a greater reward further down the line. Again, not surprising that that would be impaired, right? Antisocials, criminals are typically more impulsive, looking for immediate gratification, okay, not really looking in the long term. The other parts of the brain that were most reduced in thickness were the orbital frontal gyrus and this side of the rectal frontal gyrus, right? We can use an umbrella term for the rectal frontal gyrus and orbital frontal gyrus, which is the ventral prefrontal cortex, okay? Ventral meaning underside, okay? And that's the umbrella term for both of these subsections, okay? What does the broader research tell us that the ventral prefrontal cortex is associated with? Insight, sensitivity to others' emotional states, compassion for others, caring for others. Again, not surprising that these features would be lacking in our antisocials. Behavioral inhibition, again, we're talking about impulsive individuals. Emotion regulation, again, not surprising. These antisocials are not known for being able to regulate their emotions well. They don't make good decisions. And then response preservation is about if you continue with a behavior, even when there is the likelihood of punishment. Okay? And as we've already talked about, antisocials are more likely to go ahead unafraid of the consequences, right? In part because they don't fear condition as well. So it's meshing, right? These findings make sense in light of the broader research, right? It all goes hand in hand. It fits with what we already know. Now, initially, Adrian Rain was recruiting male participants and female participants, okay? But it was becoming a waste of time and money because, well, women are just too good, okay? They weren't scoring high enough on the antisocial measures and so on. You would have needed a very, very large sample size in order to get a group, okay, who came close to the males in terms of antisocial behaviors and psychopathy scores. Okay, not surprising, for every violent offense committed by a woman, there's 9 committed by a man. But when he went back and looked at the 12 women he had recruited, what he found was the findings were the same as the men, in that there was a negative correlation between prefrontal grey matter volume and antisocial behavior or self-reported crime. Okay, so as prefrontal cortex matter was reduced in thickness, there was the increased probability of antisocial personality disorder diagnoses and of self-reported crime for both the men and for the women. Okay, for both, there was this correlation.
Did you say they got thinner? Is that what you said?
The thinner it is, the more likely it is that they'll commit a crime, yes. Any other questions? Is everyone keeping up? Now, what's interesting is, as we've just looked at, it's the orbital region and the middle region, okay, that seem to be most connected to antisocial behavior, meaning when these two in particular are reduced in thickness, that's when there's the increased probability of crime and antisocial behavior. What we know from broader research is that these parts of the brain are thicker in women than they are in men, okay? So Adrian Rain, in a follow-up, wanted to see, since these parts of the brain are typically thicker in women, and when it is the case that they're thinner, there's an increased probability of crime, might the sex difference in thickness help explain some of the sex difference in crime rates, okay? So the first step into doing this is you have to make sure that there is a sex difference in thickness, okay? So in red, we have the women participants, in yellow, the males, okay? It's as you would expect, okay, with the broader research, this is something we know, okay? But we have to check that it's true for this sample, okay, before doing the analysis. And it is true, okay? In the female participants, the orbital region and middle region is significantly thicker, okay, than it is in the men. There's no significant difference between the inferior and superior regions, okay? Again, not surprising, we know that from the broader research, but we have to check that that it's true for this sample, okay, before we do the analysis. Again, let's check that the men have committed more crimes and are higher in antisocial personality disorder diagnoses than the women. Yes, they are. It's basically double, okay? So the red is the men, the blue is the women, okay? We have antisocial personality disorder rates and then self-reported crime, okay? The men about twice as high as the women, okay? Again, not really too surprising. But if we control for prefrontal cortex thickness, is there going to be a reduction in the sex difference? That's what we want to see, okay? So instead of just comparing all men and all women, let's just compare them when they have the same thickness of prefrontal cortex, okay? Just the men and women who have the same, okay? Let's control for that. Is this still going to be a large difference or is it going to get smaller, right? You can see here the percentage reduction in the sex difference, okay? Once we control for the thickness of the prefrontal cortex region. When we compare for the orbital and medial region, okay, the sex difference is reduced by more than 50%, okay, for both self-reported crime, which is SRC, and antisocial personality disorder diagnoses, okay. That's surprising and startling. It's extraordinary, really. This is one sample, and it's a small sample of females, okay. But the fact that in this sample you could reduce the sex difference by that much, by more than 50%, is, again, extraordinary, okay? It tells us that this very likely does have something to do, okay, with the sex differences in criminal behavior. Okay, it's at least some part of the puzzle, okay? And again, we've just looked at why that is, right? What those parts of the brain are associated with, okay? And why it might be the case that you have an increased probability of criminal behavior when they are reduced or impaired. What are your questions?
So men still showed that they committed more crimes when the sex difference was controlled for?
Right, but it was reduced by over 50%.
Okay.
Right, so this has got smaller. The difference is smaller once we control for the thickness of the prefrontal cortex. Any other questions?
It's just the gap wasn't as large between the two.
Right. So, makes sense? So I'm going to come back to talk about the prefrontal cortex on Tuesday. We'll talk about the functional differences, okay? But that's what I want to tell you in terms of the structural evidence, okay? Let me also tell you about this condition called cavum septum pellucidum, okay? Now, when you're first born, your brain will look like this picture, okay? On my right. It has this fluid-filled sac in the center of it, okay? And then what happens is during the first 6 months of life, as the limbic system is developing after birth, okay? So we're talking here about, the amygdala, hippocampus, and all these other parts of the brain that are developing. As they're developing the center of the brain around this fluid-filled sac, they're putting pressure on it from both sides, okay? Forcing it together. And within 6 months, for the vast majority of people, it then looks like this, okay? The pressure from the development of the limbic system has caused this change, okay, from this to this. For a small number of people, it takes a little bit longer, okay, but for almost everyone, within 12 months, this transformation occurs. But there's a very small number of people for whom this never occurs, and their brain always looks like this, even as an adult. We call that condition cavum septum pellucidum. Now, having this condition, what it would suggest is that something hasn't gone typical during those early stages of development, right? The limbic system must not have developed as we would expect, because if it had, the transformation would have occurred, okay? So something has gone awry, okay, during the development of the limbic system. Now, when we compare those with cavum septum pellucidum to those without the condition, there's a number of differences that emerge, okay? They're significantly higher in diagnosis rates of antisocial personality disorder. They score significantly higher on the psychopathy checklist. They have a significantly higher number of arrests and a significantly higher number of criminal convictions, okay? Based upon all these measures of antisocial behavior, okay, those with this condition score higher than those without. Now, earlier in this course, I told you that if there is a genuine connection between two variables, we should be able to see that connection from either side, okay? And we can do that with cavum septum pellucidum, because we can look for degrees of cavum septum pellucidum, okay, in comparison to antisocials versus controls. So it's not the case necessarily that your brain looks exactly like this or exactly like this. There's plenty of in between, right? It might be the case that your limbic system developed, 60% typically, okay? And so it's not really quite like this. Maybe it's a little bit more pushed together, okay? There's plenty of in between, right? It's like a zip on a fly. It's not just up or down. There's plenty of in between, right? So what we can look at is degrees of cavum septum pellucidum and have that as our dependent variable on the y-axis, okay? Instead of psychopathy or criminal convictions or whatever, we flip it around, okay? Cavum septum pellucidum is now the dependent variable. And we compare it in antisocials, the red group, and in controls, the yellow group, okay? So for example, we could look at those who have been arrested, which would be the antisocials, versus those who haven't, which would be the controls. Those with a criminal conviction, which would be the antisocials, and those who haven't, which would be the controls, okay? And again, a significantly higher degree of cavum septum pellucidum in the antisocials, okay? So from either side of looking at it, we get a significant relationship, okay? Now, having cavum septum pellucidum itself isn't going to make someone commit a crime, right? It's not really anything. It's this fluid-filled sac still in the center of your brain. What it tells us is the limbic system hasn't developed as we would expect. So we need to look closer at what that actually means. Okay, what part of the limbic system hasn't developed typically, and why might that play a part in an increased chance of criminal behavior? Before I narrow down on that, so I'm going to give you a case. Anyone heard of the case before of Charles Whitman? Well, I've told you about a number of cases, so I'm going to give you a break from my voice and I'm going to play a YouTube video on it, okay? And then it will lead on to what I want to talk about next. We are going to talk about mass killers later in the course, but obviously the part there that I'm most want you to pay attention to is the amygdala tumor brought up at the end of the video. Why might the amygdala be tied to this? Well, as it was said in the video, the amygdala is responsible for emotional responses. Fear, anxiety is crucial for fear conditioning. And a number of studies have found that the amygdala is smaller in psychopaths than in non-psychopaths, that it's structurally different. First discovered by Yang, finding it was 18% smaller in psychopaths on average than non-psychopaths, and then replicated twice in 2014 and 2019 with the same finding of 18% reduction in psychopaths in comparison to the non-psychopaths. Now, the amygdala is not just one thing, right? It's made up of substructures. And in Yang's paper, he broke it down into 13 different nuclei, okay? And the three that he found were most deformed in the psychopaths were these three here: the central nucleus, basolateral nucleus, and the cortical nucleus, okay? Now, there's pretty little research on what the individual substructures of the amygdala are associated with, because there's not very many studies that split the amygdala into substructures. But those that have looked at the central nucleus have found it's associated with stress responses, the reactivity of the autonomic nervous system, your fight or flight responses, okay? The basolateral nucleus being tied to learning from punishments, okay? So collectively to do with classical conditioning, fear conditioning especially, okay? All of which we know is impaired in psychopaths, so again, making sense. Not very much on the cortical nucleus, but one study did find that parents with a deformed cortical nucleus were more likely to be rated as bad parents by their children in comparison to controls. Okay. And again, psychopaths are not going to be the type of people who make the best parents, right? So again, that's all fitting. In a study on women, there was a negative correlation between amygdala volume, both in the right and left hemisphere, with aggressive behavior. Okay, on average, the highly aggressive women had 16 to 18% smaller amygdala, okay, than the controls. Lower left amygdala, in another study, also associated with greater likelihood of lifelong aggression. Okay. So a number of sources telling us that structural impairments for the amygdala are connected to psychopathy especially, but aggressive behavior more broadly. And again, I'm going to talk about functional evidence on Tuesday. In terms of why the amygdala might be smaller, I'm going to come to some other factors in this course, but certainly one possibility is early stress, much like the reasons that led to a shrunken hippocampus when we looked at that. Okay, some chronic stress experienced early in life. So here we have a group of controls, and then three groups, each reflecting a variable that's been tied to smaller amygdalas. So early neglect, physical abuse, poverty, okay? All of these predicted lower amygdala volume, okay, than the controls. Now, when we talked about the hippocampus is a part of the brain that's fairly fluid, fairly flexible, meaning that through cognitive activities, through memory exercises, you can actually increase the size of your hippocampus. The The best illustration of this is in London taxi drivers who have to undergo a 6-month program that's very intense, in which they have to memorize all the various different routes in London. Okay, the hippocampus actually gets larger as a consequence of this program. Okay, so the more you use it, okay, the more functional it is, the larger it gets. There's little to no evidence to suggest that the same is true for the amygdala. Okay, that this is reversible, essentially. Okay, what the evidence is telling us really is that when the amygdala is shrunken, there's nothing that can really be done to get it back to regular size. Okay, so it's a more permanent effect. And again, behavioral problems associated with reduced amygdala volume. What are your questions on what I've covered today?